So what we're going to do now is move back into the Unit 3 area of Study 1. And you've got a lot of information about DNA and protein synthesis and things like that. Taking that concept of what a gene is now moving through how genes function. How does this protein synthesis occur? When does it occur? So that first and one of the most key questions of what is a gene is one of the key questions as to how do cells function. And of course, the answer to that question is it is a section of DNA that codes for a protein for a particular cell function or characteristic. Nice full definition of what a gene is. And by the way, I'm just on page 64 in our book. So that's the idea of what a gene is. Scientists, once finding that, then did a lot of investigation, knocking out genes, changing, mutating DNA, using computers to realize there's actually 3 different types of gene, depending on what the protein that they code for actually does. Now, the first type, the one we tend to talk about mostly, are what we call structural genes. And these are the genes that code for enzymes, they code for protein structures within the cell, and they are the ones that give you the colour of your hair, that give you the colour of your skin and eyes and height and all those. They're the structural genes. And down further here, it's a gene that codes for protein involved in cell function. So they are the structural genes. It's these ones that gives the different features of cells, which is called morphological. So it's these genes that makes the skin cell look different to a hair cell, looks different to a muscle cell. The proteins they code for are the ones that control that type of thing, the structural gene. And that's what we're particularly used for. Okay, then in investigating the genomes of animals, remember a genome is the full set of genes for a particular species, and in particular the human genome, they realise most of the DNA is in fact not structural genes. When they thought there was going to be over 100,000 genes coding for enzymes and that, they only worked out there's about 25,000 structural genes in the human genome. So then they said, well, what's all the rest of the DNA about? Well, most of the DNA is in fact regulatory genes. They are still sections of DNA. They still code for a protein. But what regulatory genes do is they control the expression of other genes. These are the ones that are going to turn off or on other genes. So what they do is they code for a protein, and we call those proteins transcription factors. And an example that we particularly talk about is a repressor protein. That is one of the key types of transcription factors these genes code for. So regulatory genes code for a protein. That controls, by turning on or off, other genes. Now, there is a third type of gene, which we used to learn a lot in the old course, and it's quite complex information. So I'm just going to give it here. They're called homeotic genes or master genes. just so that you're aware that they exist. And when you do Unit 4 and talk about species, it's homeotic genes that determine the different species. And they code for proteins, which is what all genes do, for development of the embryo and of the species. So when you talk about evolution and species and all that, it's these third class of genes that have altered to make the different species. But for our purpose at the moment, we're just going to be talking about this relationship between the structural gene and the regulatory gene, which means this is what gene regulation is all about. Now, I'm not sure how much you know about going from that very first cell that formed you into what you are now. But when the egg and sperm joined, they made that very first cell. We call it the zygote. That's the first cell of a living organism. That cell has the full set of its DNA, half from mum, half from dad, but it's got the full set in it. From that point on, that cell undergoes cell division. Copying, absolute copying. It's called mitosis. And we get this ball of cells called a blastocyst or stem cells. Every single one of those cells contains exactly the same DNA. It's just a copying process. So therefore, every cell of your body has exactly the same DNA. The genome, the set of genes, is the same in every cell of your body. Then as progression happens, the cells through the homeotic genes, but then also other genes, you start to become what is recognizable as an embryo. Skin starts to form as skin cells, muscle starts to form as muscle cells. And yet every cell of your body has exactly the same genes, but not every gene is turned on. So in some cells, most genes never turn on. For example, in skin cells. In skin cells, there's muscle genes, brain genes, teeth genes. Every single gene is in your skin cells. But only the skin genes get turned on. And the others are permanently turned off, locked off. This is part of gene regulation. Being able to turn on genes and have some permanently closed off. Also, what they found was that in any one particular cell, the genes aren't switched on all the time. It's really important to have this regulation of the genes, turning them on and off. I equate it to the light switch. When you leave your room, you turn the lights off. When you come back in, you turn them on. So you only turn the lights on when it's needed. Not during the day, only at night, whatever. Now, the reason you're doing that, hopefully because your parents have asked you to, but it saves energy. Well, same with this. By having genes turned off when they're not needed means that the cell isn't wasting energy. Turning on transcription, translation, protein synthesis is a high energy process. It uses a lot of energy called ATP. So for regulation of genes, what we're going to do is save the cell energy, save the organism energy wastage. Now you might think, so what? Well, we're limited in how much energy is in a cell at any one time. We don't eat constantly. And sometimes organisms might need a burst of energy to escape a predator or something like that. Or it might be that a saving of energy is enough just to prolong an organism's life to get to reproductive age, to keep their species going. So energy saving is really important. It also saves resources. To make proteins, you need amino acids. Again, there's a limited supply of amino acids in the cytosol. And again, it needs energy to do this, making a protein. So to have ATP, you need the resources to make the ATP. So by turning the lights off saves energy, but it also saves electricity, the resources. So this gene regulation is really quite an important thing. And you might think us being humans, the top of the evolutionary chain, yeah, I guess we are, that we have got this gene regulation really absolutely perfect. And we do. We have very complicated gene regulation processes. But what is fascinating is gene regulation even happens in bacteria. The most simple of the evolutionary organisms, single-celled prokaryotic bacteria, undergo gene regulation. And in fact, it's the bacteria in particular that has been studied by scientists to really try to understand how this turning on and turning off of genes happens. So what we do in our course is we don't look at eukaryotic gene regulation. It's far too complicated, but we do want you to have a bit of an understanding of just this overview of how gene regulation occurs. We use a bacterial example, but to equate it to eukaryotic cells is not too difficult to do as well. So gene regulation in prokaryotic cells. Now, on page 47 in our book, you might recognize this diagram. This is a piece of DNA, and we have a section of the DNA called the promoter region. And then we have the coding region. And then we have the terminator region. And then we have the rest of the DNA. And in our books earlier, this was the gene. The coding region was the part that had the start triplet, like TAC is the start triplet, and it had a stop triplet, and it had the introns and exons, etc. So that's what we learned as a gene. And we learned, I hope you remember, that this is where the enzyme for transcription, called RNA polymerase, joins onto the start of the gene, the promoter region. It moves along, transcribing as it goes, forming the, I'm just trying to find a color, orange, forming the pre-mRNA. And that is transcription. And then when the RNA polymerase gets to this end, it detaches because it's finished its transcription. So that's the process of transcription, if you like. Now, through science and all the workings of it, especially with bacteria, what scientists have worked out, there's, oh, didn't mean to do that bit. There's actually another section in bacteria. I'll just put my start there. And this section is called the operator region. And in bacteria, each gene or set of genes has an operator in front. In eukaryotic cells, they're not sure if each gene has its own operator region or how it works. It's much more complex, but they have studied bacterial genes. So what we can do is understand that to turn a gene off means to stop transcription. To stop transcription means to stop the RNA polymerase moving along. To turn a gene on means to let the RNA polymerase move along and do transcription. And it's as simple as that. But how does this actually happen? What stops the RNA polymerase? and what allows the RNA polymerase to move along. Well, the example that we're learning is called the TRP Operon. Now, this is the new course. Starting this year, we've got the updated course. In the previous course, there was a similar kind of thing called the LAC Operon. The LAC Operon is no longer the part of the course we're doing. It looks a very similar process, but they've changed it in the new course to be the TRP Operon. So please, your teachers might still explain the LAC Operon to you, and that's absolutely fine because it's another example of gene regulation, but it is the TRP Operon that you'll be asked about in exams. All right, so here's a few little bits of word. What is an operon? Now, in my picture here, I've got a gene with the promoter, operator, gene, terminator. That is not an operon. An operon is where you have a cluster of genes under the control of a single promoter region. So if you like, in our picture, there's the promoter region, there's the operator region. We've got a series of genes under that same operator region. This is an operon. So it's not just a single gene, it's a cluster of genes. Now, the cluster of genes they were looking at in the TRP operon, there's actually 5 of them. There are 5 genes in sequence, and they are all controlled by the single promoter and the single operator region. Now, TRP. What does TRP stand for? It stands for tryptophan. And tryptophan is an amino acid. It's an essential amino acid in a lot of proteins, but it is absolutely essential by bacteria, in particular E. coli. Now, bacteria cannot make tryptophan all the time. It takes it up from the environment when it's there. So it will absorb it whenever tryptophan is in the environment the bacteria is growing in. But if a bacteria is in an environment that doesn't have tryptophan, it still needs it. That's when it needs to make it. That's when it needs to turn on its tryptophan-making genes. So the mechanism, as it says here, of this operon. The operon, which is the series of genes, is activated when tryptophan is not available. And this is how the bacteria can make its own. So that is turning the gene on. Activation means turning it on. That means transcription's happening. But as I said, when tryptophan is available in the environment, the bacteria doesn't want all these enzymes being made. So the bacteria turns its operon off. The tryptophan operon is repressed or turned off. And the E. coli takes up tryptophan from the environment. So it doesn't need to have wasting all that energy in making its own tryptophan because it only needs what it needs. So to look at how this on and off scenario works is to understand these sort of pictures, if you like, here. All right, so first of all, the first scenario is when we have, what colour was I using? I want to use the blue. The first scenario is when tryptophan is present. In other words, we want the operon turned off. Okay, so here is the bacterial DNA. And bacterial DNA is actually a circular piece of DNA. So what we're talking about is a section of the DNA, and there would be this set of genes there. This here is the operon. This section here is the operon. So we would have our series of 5 enzyme genes here. Here's our promoter region. Here's our operator region. And here are our genes. So that's the operon, that part of the picture. Now, elsewhere, let's say here on the bacterial, is the regulatory gene, which is this one here. So this is the regulatory gene, and this in our picture is the operon. So the regulatory gene is a code for a protein, and the protein it codes for is called a repressor protein. So this is the repressor protein. Notice it has a shape. All proteins have shape. Notice the shape here, this bit here, does not match the operator region. It's sort of got its little sticks, not quite matching. Okay, so when tryptophan is present, here's some tryptophan. The tryptophan will join onto the repressor protein. So when tryptophan is present in the bacteria, which means it's in the environment, it's moved into the bacteria, it attaches to the repressor protein. And what it does, it then activates it, changing its shape so that it fits the operator sequence. So the repressor protein changes shape. Because the tryptophan has joined it. And it joins onto, not the promoter region, onto the operator region. So here we have our repressor protein attached to the operator. And if you have a look, the RNA polymerase, which is this grey bubble, which is on the promoter region, it cannot move across to transcribe the genes. The repressor protein has blocked its pathway. So if the repressor protein is joined to the operator region, that pathway is blocked, and so the RNA polymerase cannot move across. So the genes aren't transcribed. The enzymes aren't made. But they don't need to be because the bacteria has the tryptophan. So that is how the repressor protein blocks the operator when tryptophan is present. Okay, so the next scenario. What if there's no tryptophan? If there's no tryptophan in the environment, that means there's no tryptophan in the bacteria cell. So the bacteria has to make its own tryptophan. To do that, it needs those 5 enzymes. It needs the 5 genes transcribed and translated to make the 5 enzymes. So this is when we want the operon to be turned on. We don't want the repressor protein blocking it. So here again is our gene regulation, and here's our repressor protein. Just like before, it's still being made. But now there's no tryptophan to change its shape. So it is not able to fit and block the operator region. Because it doesn't fit. The shape's wrong. Which means the RNA polymerase, which is joined to the promoter, can now move along, transcribe each of the genes, TRP A, B, C, D, and E, we'll just call them that, into the mRNA. Then each of those will be translated into the specific enzyme. And these 5 enzymes that have now been made will produce tryptophan. They're needed to activate the tryptophan reaction. They don't make tryptophan, but they're the enzymes that will control that tryptophan reaction. So here we have an example of understanding what a gene is: promoter, coding region, terminator, but also understanding there's this section called the operator. And if a repressor protein blocks the operator, transcription can't happen. If the repressor protein doesn't block, transcription will. And in the tryptophan operon, we have, when tryptophan is present, it joins to the repressor protein, and the repressor protein can do the blocking. When tryptophan isn't present, it doesn't fit. So it can't block, so transcription will happen. An ingenious way of saving energy, saving resources, doing this turn on, turn off as the bacteria needs tryptophan. And as you can imagine, as tryptophan is being made, there's my tryptophan, when there's more than enough made, it will then join onto the repressor protein and the operon will turn off. And so the bacteria will use up its tryptophan, and if it doesn't get any more, then the tryptophan will be used that was attached to the repressor, and the repressor will come off. So it's a constant on and off scenario. If a bacteria has tryptophan in the environment all the time, it's never turned on. But there are often situations where the amount of tryptophan may not be adequate for what the bacteria needs. So it might just need to turn it on for a little while, make its tryptophan, and then turn off when it's got enough. Gene regulation. And that's how genes are turned on in cells to make whatever cell it's going to be. And once turned on, it might then be turned off permanently because that cell has then formed and functioned as it needs to. So the world of taking the concept of transcription, translation, protein synthesis, nucleic acids, proteins, all that work that you did with Veronica, taking it into the world of how scientists have gone further than just understanding a process. They're now understanding how these genes work and how they're regulated by these other regulatory genes. But Damien, your question, is the promoter part of the operon? By definition, the operon is the cluster of genes and the promoter as well. So this whole thing would be seen as the TRP operon. And how many genes? Well, there's actually meant to be 5 genes in the picture, but so it isn't just the genes, but if you have a single gene and a promoter, that's not an operon. So the promoter is still part of the gene, just like this picture here. The promoter is part of the gene. So when you have a cluster of genes with just that single promoter and operator in front, that's what the operon is. So the operon is the whole section of that DNA. As I showed, when I circled this here, I'm circling the whole operon. So it's promoter, operator, and the cluster of genes that are involved as well. And what amazes me is, I mean, you can't see these DNA sequences and things like that. So it's obviously all done with incredible skill and computerization and all that. The whole world of genetic technology is quite extraordinary and has only been around. DNA knowledge of understanding of its structure has only been around for just over 50 years. So it's quite mind-blowing that we can get all this information from in such a short time. Okay, oh, great, Damien. What does tryptophan actually do? Tryptophan is one of the amino acids. There are 20 different amino acids in all of nature. Tryptophan is one of them. In bacteria, it is an essential one. Most of the bacterial proteins do have tryptophan in it, and some of the key proteins that are involved in bacterial function and in our proteins as well. It's a very important amino acid. They are all important, but tryptophan is the one that they've been able to study in bacteria. As I said, last year we did the lac operon, which was about lactose digestion by bacteria, and lactose is a sugar. So their operons aren't just about to make amino acids, they are also about to make and do other processes in cells as well. So the world of genes, what they are, how they work, and now how they are regulated.